Okay, so it looks like it's about time to start. Seems like it's day before Golden Week, so there's some people who are not here, but it's okay. Since we have a pretty small class, I think it's good. It's possible for me to remember everybody's name, so I think I'm going to try to learn everybody's name from today. And it's good that there are some people who aren't here, but actually they told me they're not here, and so I have their emails, so I know who they are now. But now I need to call some attendants to see who is here. And I guess in this class I will just use people's last name, just family name. So I will call, for instance, Tsukasa-san. Is Tsukasa-san here? Okay, so you're Tsukasa Yuma-san, right? Yeah, okay. I have you for Rigaku, your second year. Are you in Rigaku? Yes. Your science, okay. Alright, so then next is Toriba-san.
Hi.
Okay. Yeah, so first year in medicine, School of Medicine, right? And then I have, so I think your family name is Wu. Is Wu here? Yeah, okay. Is it? What should I call you in this class? What do you want me to? Should I call you Wu-san? Wu-san is fine. Okay. Wu-san is here. And you're fourth year Nogaku. Right. Okay. And then I have Makata-san. Yes. Yeah, I got your mail from last time. And then Fujita-san. Yep, okay. I think I already know. But I don't know if I'll be able to just look at you and remember. And then Anna, is that all right? Should I call you Anna or Emerson-san?
Just sounds good.
Anna is fine. Okay. That's good. Okay. So, all right, that's pretty good. So yeah, now we can get started. And the topic of today's lecture is what chromosomes do during cell division and how chromosomes are able to make it into the daughter cells. And along the way, of course, we have to learn a little bit about cell division process itself, because it's not all about the chromosomes. The chromosomes have to coordinate with other things that are going on inside the cell. All the lights off because there's going to be some movies. But if I draw on the board like this, I just want everybody to, if I write like this big, can everybody see that okay? Is that okay for everybody? If you're sitting way on that side of the classroom, then it may be difficult for you to see through this sometimes. So maybe from next time, it would be good if more people were just over on this side. Because if I write things on the board, it's generally going to be in this area here. So as long as you make sure you can see what's going on here, it should be okay. All right, so I'm going to show you, first of all, I like to try to put in some movies so you can get excited about the process of cell division here. This is one of the most striking examples of synchronized cell division that's known. This is an embryo of Shoujobai, the Drosophila, and what you can see here is that it's already gone through many cell divisions. So this is way after the first cell division. There's probably already 1,000 cells or so in this embryo. But right now we're looking at just the top surface of the embryo. And each thing that you see here, I hope you can realize that these are nuclei here. These are the cell nuclei. But they're fluorescent, and the reason they're fluorescent, the reason we can see them in a microscope, is because this Drosophila embryo has been injected with a fluorescent protein. So, can anybody guess what protein was injected into them that would enable you to see the DNA? It's a protein we've learned about. Actually, it's a histone protein. So people took the purified histone proteins and then added a fluorescent tag to each histone molecule and then injected it into this Drosophila embryo. And then histones behave just like normal histones. They get wrapped up in the DNA. So you can see, of course, there's a lot of histones that are not in the DNA, and that's why we see some kind of fuzzy background fluorescence in between the nuclei. But when they get wrapped up by chromosomes, these fluorescent histones get packed really close together, and that's why we can see these big nuclei here. And what you can see as time goes on, they change, right? So it happened really quickly, but we went from these nice round nuclei now to everything is in kind of a line, and you can tell that all these lines are kind of oriented in the same direction. You can't see very well, but actually each line contains 8 chromosomes in it that are about ready to undergo a division. Now you saw that they all divided there. And they all divided at the same time, which is really a kind of unique thing that's happening in this. And I'll let it go for a few more times. Each time one of these cell divisions happens, of course, the number of cells in the embryo doubles. So it was going from, well, it's a huge number already, probably now tens of thousands of cells. Now there's probably, eventually there'll be hundreds of thousands of cells. And you can see that they all maintain this really tight synchrony as embryonic development proceeds. It goes like this for another few cell divisions, and then they start getting out of sync and the different tissues start forming. But up until now, what you've seen is a very regular process where every single cell, every nucleus seems to be on this synchronized clock where it's doing a certain order of events. And you've probably heard, you remember this from early biology classes, that all of these cells are doing this repetitive cycle of events called the cell cycle. And the cell cycle is best understood as being composed of these 4 parts. But it's really, for our purposes today, there's really 2 parts. So one group of these first 3 phases here are what's known as interphase. And these are, they're actually called G1 phase and the S phase and the G2 phase. And G stands for growth, and it's not really, even though it's not really an exciting dynamic phase as far as chromosome movement or chromosome dynamics goes, it's actually, of course, really important because during this stage, the cells are growing and making all the materials they need in order to make a full-sized copy of themselves. So when cells divide, of course, there needs to be enough material to make a full copy of that cell. The reason why there's a separate S phase as opposed to these 2 other growth phases, the cell is growing this entire time, but something special is happening in the S phase, and that is when the DNA itself is replicating. And that takes, so to replicate DNA takes enough special resources of the cell that an entire different cell cycle stage is dedicated to it. So in G1, there's synthesis and growth of things that are needed to synthesize DNA. Then in S phase, almost all the cell resources are devoted to replicating the chromosomes. And then there's another final bit of growth. Some cells actually don't really have a G2 phase. They go directly from the S phase to the M phase. But then it's mitosis is what we're going to be concerned with in this class. So we're not going to spend much time in interphase other than to remind you of what this looks like. I'm going to show some pictures of this, of cells that are staying like this. This is not fluorescent staining. This is the kind of stain that people might have used 100 years ago, but it's still quite useful to visualize the different cell cycle stages. And most of the cells you see at any time are in interphase, where cells are growing. And you can see that to get from this cell to this cell, a lot of growth has occurred. So there's definitely, not only do the chromosomes need to replicate, but the entire cell needs to grow in order to make daughter cells like that. Okay, so we're interested in mitosis, but of course mitosis is itself a very complicated multi-step process. Mitosis isn't just one thing that happens. Today we're going to kind of go into these mitotic substages, and there's going to be, it may be new vocabulary for some people, so I'll try to slow down and say things repetitively so you can remember some of these words. And where there's a word that I think is, I've tried to underline words that may be maybe unfamiliar or technical. So we'll try to go through these words. If I say a word that you didn't catch or something is unfamiliar, please go ahead and stop me, and I can always re-explain. I'm by no means committed to just standing up here and talking for an hour and a half straight. I would love if somebody could jump in and interrupt me. So please feel free to do that at any time. So mitosis starts. So the difference between this and this, you can see quite clearly with your eyes, is that chromosomes have to start condensing. And we went over a lot about how chromosomes do this condensation last time. So if you remember, first of all, chromosomes are wrapped around histones, and then these histones may or may not be packed together in a heterochromatin state to get some condensation. But then to really condense, to get from this open, kind of chaotic-looking interphase morphology to the tight-packed mitotic chromosome morphology, they need to do this, what's known as condensation, and these proteins, these condensing proteins, are what help chromosomes condense down to that packed state. So chromosomes that have a lot of these condensins on them, they extrude these loops out of them, and eventually, if enough loops are formed over the entire chromosome, it'll shrink down into a condensed state. And I found a really cool simulation of this. So if you keep in mind this kind of interphase chromosome structure, there's a group that did a computer simulation. So this should represent one chromosome that's replicated into two sister chromatids. So I'm kind of skipping over the process of chromosome replication for now, but you have to keep in mind that before mitosis, there's the S phase. So a single chromosome gets replicated by these kind of replication forks growing. And eventually there's eventually two chromosomes. But as we'll see later, it's kind of tricky for these two chromosomes to get separated. And so the chromosomes in G2 or in interphase are actually replicated. So there's one, let's say there's one red and one blue chromosome, like there is in this movie. And yet they can kind of condense into a structure where the red chromosome is on one side and the blue chromosome is on one side, even though they start out all tangled up. And just this process of extruding these loops is enough to do it. So each little kind of blob you see, these are 16 independent simulations that end up at the same same endpoint. You can see that the surface of these writhing balls of chromatin are these loops that are getting extruded. And in each case, what started as a tangled ball of chromosomes ends up getting resolved into two nicely separated replicated chromosomes. Here they're connected by by the center part of the chromosome, or the centromere. Just some artificial connection has been imposed here at the centromere. And so you can see that even though chromosomes start out in interphase where they're all kind of tangled together, this process that that I've showed you, where loops come out and pull chromosomes together, are able to separate into what looks pretty much like mitotic chromosomes. Okay, and so this is what is happening to get to this morphology. And so this is what's known as prophase, and it's the first stage of mitosis. So to get from here to here, these chromosomes are doing this loop extrusion, and each chromosome is contracting and separating from the other chromosomes in the same nucleus. And here I'm finally telling you which organism this is. This is just a bean. These are roots from a bean. And they happen to have very nice big chromosomes. So yeah, if you look closely at this prophase chromosome, you can see some cases where the two sisters have come apart. And just for some nomenclature here, some words. This, of course, is called a chromosome, and it's one long DNA molecule. When it's replicated into two strands, it's also called a chromosome. But now each separate strand has a different word, and these are now called chromatids. Or sister chromatids. So sister chromatids. So if I use the word chromatid, it means one of the two replicated DNA molecules that make up an entire chromosome. So even before or after DNA replication, they're both called chromosomes. But a replicated chromosome in late S phase, or in G2 phase, or in prophase, consists of two sister chromatids. So you can see the chromatids here kind of separate from each other, but mostly they're still really kind of packed next to each other, held together with cohesin, so that you don't see the two strands. The other thing to notice about this stage of prophase is that even though some space has opened up between these chromosomes, it's still in this very nice spherical shape, right? If you compare it with something like this, where chromosomes have gone all the way out to the edge of the cell, here the chromosomes are still fairly far away from the edge of the cell. So can somebody tell me why don't these chromosomes go out to the edge of the cell? Can you tell me why chromosomes don't go all the way out? Are you translating your answer? Anybody?
Covered by membrane?
Mm-hmm. Yeah, yes. Right, so there's a membrane called the nuclear membrane. That, just like these interphase chromosomes, they're also enclosed inside this membrane. So the chromosomes have started to condense, but there's still this membrane that is keeping them there. And let's see. Right, so we're going to the next step now. So the first thing that happens is this nuclear membrane, or nuclear envelope as it's called, breaks down. Okay. And then, well, let's do one thing at a time. So I should talk a little bit about this nuclear membrane because it's quite important. So in interphase, I'll draw all the chromosomes like this. You've just got a bunch of individual chromosomes that are not condensed yet. So they're each kind of in their own little area. And they're surrounded by this nuclear membrane. So we can call it the nuclear membrane. Or nuclear envelope, either one is fine. But actually, envelope is a little bit preferable, and that's because membrane usually refers to one film, like one soap bubble film of a membrane. But in the case of the nucleus, there's actually two, right? You may remember this from high school. So there's actually two nuclear membranes, or a double nuclear membrane, that together make up what's called the nuclear envelope. And this nuclear envelope is not, if it just surrounded the DNA, of course, then the DNA would not be able to connect to the outside world. So there are actually thousands of little holes in this nuclear membrane called nuclear pores that have some proteins that allow things to come in and out. So this is a nuclear pore. Pore just kind of means a hole in the nucleus. And then at the sites of nuclear pores, actually this nuclear envelope kind of connects to itself like this, bends around and connects to itself. So the nuclear envelope is really made of two nuclear membranes, but these holes allow things that are on this side to come around and go to this side. So it's really kind of one. It's really not a double membrane. There's really two. It's really one membrane, but it's in this kind of double structure. Okay, and all the DNA is in here. And at the very first stage, you get this condensation of DNA into prophase. But for the DNA to divide into opposite cells, this nuclear membrane is kind of in the way, right? You can't, in a large cell like these beans or in our cells, the nuclear envelope would prevent chromosomes from going to the opposite poles. So we have to get rid of it. Okay. So let's see. Yeah. Anyway, I'll show a little more about getting rid of it. So that's the first thing that has to happen. This nuclear membrane has to go away. Secondly, the nuclear membrane breaks down, and then the chromosome kinetochores get captured by the spindle. And those words may be a little bit unfamiliar, so I'll talk about what those are. And then finally, once they're captured, chromosomes move to the center of the cell. So that's a lot happening in mitosis. So I'll remind you here that chromosomes have this specialized part called the centromere, and the job of the centromere, which is the DNA sequence, its job is to recruit something called the kinetochore, which is a protein machine. I'm referring to it as the handle on a chromosome, so a cell can pull on this kinetochore. But as you'll see later, it's not just a handle. It's not just an inert blob that the cell can pull on. It's actually a really complicated machine that is involved in sending signals. So we shouldn't think of the kinetochore as just a handle that the cell pulls on. But for the purposes of metaphase and what the cell is doing, for now we can think of it as just a handle. Okay, so I've explained nuclear envelope, and I reminded you what kinetochores are, I have to tell you what is the spindle now, and that is this thing. So the spindle is the green structure that you see in this cell, and that this stage here is also metaphase. And these chromosomes are in the process of getting captured. Probably most of them are already captured by this spindle. So first of all, spindle is just is this kind of object, and it's used to wrap when you're spinning thread. When you're making yarn, you spin it around this, and it tends to make this kind of shape where it's kind of thick in the middle and thin at the edges. And it's called bosui or spindle just because it has this kind of shape. So it has nothing to do with actual string. It's just this kind of shape. When people first saw the spindle shape, it reminded them of this because it was still the 19th century, and probably this was a very familiar object to a lot of these people. Now, if we saw what that was, I don't know if too many people would recognize what it is. But back then, they named things over things after things that were familiar to them. So this is the spindle. But what is it? Okay, I have to kind of explain not only the shape, but what is it. So if anybody already knows, do you know, so you see all these green threads coming out. Does anybody know what those threads are?
Microtubule.
Mm-hmm. Yeah, yes, those microtubules, right. And so each thing we're seeing here is a microtubule. Okay, and the spindle is made simply of microtubules. Okay, that's one part of the spindle. So you've got these microtubules, and it looks like they're all coming out. They're kind of coming out from here and from here, right? So there's this structure here called the centrosome. And it's the job of the centrosome to be a source of microtubule growth. Okay. So microtubules are not just simple threads. Like DNA, microtubules also have an orientation. So they're polymers that have a certain shape to them, and this shape gives them these two different ends. So they have what's called a plus end and a minus end, right? And the minus ends come out from the centrosome, and they grow. Microtubules grow toward the plus end, and they grow by adding these tiny subunits of protein called tubulin. So tubulin molecules can add to the end of microtubules to make it grow. At some point, though, the ends of microtubules are quite unstable. So after growing for a while, microtubules will always shrink back. So if you look at a movie of microtubules, you'll find that they're always growing and shrinking and growing and shrinking. And that's just a property that is built into microtubules. So tubulin subunits can add on, but at some point they stop adding on, and then it becomes very unstable, and then it all contracts back. So if you look over time, for instance, at the length of a microtubule, it will grow and grow and grow, and then immediately it will shrink, and the shrinking is faster than the growing. So it may grow longer the next time, and then finally it shrinks again, and then maybe it'll start growing again like this. And all the microtubules are doing this at the same time. So this spindle structure that you see, even though it looks like this kind of stable structure, it's actually full of these dynamically growing and shrinking microtubules. And the reason, at least one of the reasons why they need to grow and shrink, is because each kinetochore, which are shown by these red dots here, each kinetochore needs to encounter the end of a growing microtubule. Okay, so the chromosome is sitting here somewhere in the cell, the nuclear envelope has broken down. Okay, here's chromosome, and here's its kinetochores, like this. But it's just sitting in the cell, floating around, and it needs to divide, right? It needs to go to the daughter cells. In order for that to happen, it needs to first come into the center. Yeah, there's actually another centrosome here. There's another microtubule coming from here. So this chromosome that's sitting out here, somewhere in the cell, needs to be brought to the middle because it's going to be pulled apart by this spindle. So the way it gets brought there is eventually, after many cycles of growing and shrinking, eventually randomly one microtubule will find the kinetochore. And when it does, it actually binds to it. Then during the shrinking process, it can pull the chromosome down toward it. So as it grows, it may eventually encounter a chromosome, and finally it will shrink back and be able to pull the chromosome somewhere in the middle of the spindle like this. But that's not enough, right? It can get pulled by one spindle, but it has to be captured by the entire spindle. So eventually, another microtubule from this spindle has to connect to it. And then finally, the chromosome will end up in the middle, like this. Okay. So the chromosomes start floating out somewhere in the cell, and eventually, by the growing and shrinking of this dynamic structure, the mitotic spindle, eventually it randomly gets captured by a microtubule, and then captured by another microtubule. So that's a lot of randomness that's happening. But eventually, with time, eventually all the chromosomes will get caught, and then you end up with chromosomes that are in metaphase. So when all the chromosomes, or as chromosomes are moving to the center, and then they spend a little time hanging out at the center, this is what's known as metaphase. You can see that they're all at the center because you've got this kind of line here of, what do you say, this, I don't know if it's obvious to you that this is where the chromosomes are getting pulled equally from microtubules coming from this side and microtubules coming from this side. So it's much easier to see here, right? This is much easier to see that all the chromosomes have come to the center. And in fact, if you look at these kinetochores, so each red dot here is a kinetochore, you can see on each chromosome, the kinetochores are in pairs and they're facing away from each other, parallel to the direction of these spindle microtubules. And that shows you, this is part of the evidence that show definitely that each chromosome is getting pulled by microtubules from opposite sides. And we can see it really well on this one, actually. This red kinetochore is being pulled probably by that microtubule, and this same red kinetochore that's paired with it is pulled by the other one. You can't see it so well where things are crowded, but here at the edge, you can see quite well that this chromosome is getting pulled apart. And the fact that these kinetochores are apart from each other shows that actually they're feeling some force from the spindle, and that force is really important for the timing that we'll see later. Okay, so after they've spent some time, then eventually the chromosomes do what they've been waiting for, and that is they divide. And that's the next stage of mitosis, which is anaphase. And anaphase, there can be two different kinds of anaphase depending on how the spindle is constructed. And so conceptually, anaphase can either be, well, some combination of anaphase A, which is that eventually chromosomes come apart. And just as was happening with the dynamic movement of growth and shrinking of microtubules to capture the chromosomes, now when the cell is dividing, this microtubule gradually shrinks and pulls the chromosome with it as it's shrinking. You can see this is maybe time 0, and then with time, it slowly coming back and it's pulling the chromosome with it. So you might wonder if the microtubule is pushing against the chromosome and it's shrinking by losing tubulin from the end, how is it able to pull the chromosome if it's losing these tubulin proteins at the end, right? This is something that always bothered me when I was doing this. So this microtubule, say, is made of a bunch of individual tubulin proteins, right? So each one is a tubulin protein. And now it grows by adding tubulin proteins and it shrinks by losing tubulin proteins, right? And that's the only way these things can grow or shrink is by adding or losing individual proteins. So if the microtubule is attached to the kinetic core of the chromosome like this, and it can pull the chromosome by losing subunits, how does that work? Because as soon as it loses this subunit, it's not attached anymore, right? And then isn't this microtubule just going to come away? Is there any? You think there's some kind of Pac-Man mechanism where this is kind of chewing like this? Yeah, that's basically right. Yeah. So it just means that there's more connection going on between the microtubule and the kinetic core than just at the very end. There's actually the connection is kind of more like this, where yeah, you're right. It is kind of like a Pac-Man. So the kinetic core is a lot bigger than just touching the last microtubule subunits. So it's not grabbing just here, but it's really grabbing more like here. And then as this shrinks a little bit, the kinetic core attachment is free to slide up and down a little bit on the microtubule. So it's not just grabbing tightly here, but it's kind of moving up and down. So as this gets shorter, the kinetic core is always able to grab the next subunit and the next subunit. So yeah, exactly, it's kind of a Pac-Man mechanism. This is what happens in anaphase A. And so anaphase A is kind of the dominant type of anaphase in an organism with a small genome. And a classic organism with a small genome that does anaphase A almost exclusively is budding yeast. I made this two-frame animation that I hope you can tell. You can kind of see what's happening here. So the microtubule is connected to the chromosome like that, and the microtubule just gets shorter. It gets shorter, and as it gets shorter, it pulls the chromosome through. Now, what I've drawn in red here is the nuclear envelope in yeast. And you might wonder why is a nuclear envelope there. I thought the nuclear envelope was supposed to break down. But actually, in many organisms, the nuclear envelope doesn't actually break down. And budding yeast is one of those organisms. So somehow the microtubules are able to reach in. The microtubules actually start inside the nucleus. They're not outside the nucleus. They're inside the whole time. So that's actually kind of wrong. This thing should be inside the nucleus. Sorry about that. But the microtubules are always inside the nucleus, and the whole nucleus gets elongated and finally breaks in half. Okay, so that's anaphase A, whereas anaphase B is something completely different. Anaphase A relies on shrinking the microtubules, losing subunits, and pulling the chromosome, so the distance between the chromosome and the pole gets smaller. Anaphase B, however, the entire spindle actually gets bigger. So instead of microtubule shrinkage, anaphase B relies on microtubules growing. As these microtubules grow and get bigger and bigger, there's a force that actually pushes both spindle poles apart. So the centrosomes move apart from each other. And that's something you can see very well.
Oh.
Let me do. maybe I can turn this around. Oh dear. Yeah. That's moving up good. There we go. That's a little bit better. It goes kind of fast. I hope what you can see is that, so here's one pole and here's the other pole, and they should move apart from each other. And this is the very first cell division that's happening in this embryo, so it's got the biggest, most dramatic, oh dear. Sure, I think you can get that. It looked fine on my other computer, but I think it got really compressed here. Sorry about that. Anyway, the next picture is a little better. So after doing that 7 times, this is what the same sea urchin embryo looks like. Now it's got a ton of metaphase chromosomes in it. Right. So yeah. So now back to our humble solanine, this fava beans. Can anyone? You all know which one is the anaphase now, right? Yes. So you just have to imagine that that this spindle has grown, and it's clearly. I don't know if you were to measure the distance. If this is a metaphase spindle, and this is an anaphase spindle. I think it's probably longer. So there's probably some anaphase B. But in organisms like us and beings, there's actually both anaphase A and anaphase B happening. So we're simultaneously pulling chromosomes back by shrinking these microtubules, and at the same time, other microtubules are growing, and so the distance between the spindles is increasing. And that means actually that there's two different kinds of microtubules, and they're both doing different things. Okay, so the ones that are attached to the kinetochore are actually called kinetochore microtubules. And microtubules that are not attached to anything are still very important because they make up the majority of this spindle. So these are the spindle microtubules. And does anybody know how? So these microtubules grow. Is that enough to push apart? If this is one microtubule and this is another microtubule and it's growing, is that enough to push the poles apart? I don't think so. So just because they're growing doesn't mean that's not enough force actually to push them apart. Something else has to be working on these microtubules. And in fact, there's a large class of what are known as motor proteins. So I have to show what's going on here. There's these things here called motor proteins. And what a motor protein does is it likes to move along the microtubule. So it can be bound to one microtubule and move along the other microtubule. So it's getting a little crowded here, but let's just show 2 microtubules here, and remember the minus ends are here and plus ends are here. So if you have a motor protein that's bound to one microtubule, and they actually have legs, sort of, and these legs will walk along another microtubule. If this part is bound really tightly and it doesn't move, but these legs can walk along another microtubule, then can you see which way does this motor protein have to walk in order to make the spindle grow? So motor proteins are divided into minus end and plus end based on the way they like to walk. So with this spindle extending motor protein via plus end or a minus end?
Plus end.
Yes. That's right. Yeah. So if this is stuck on here really tightly, and this likes to move to the plus end, then it will take its next step toward the plus end, and that will make a force, very small force, but enough opposite force that will push this part backwards. And likewise, that will exert a force here that will push it that way. And likewise, there's probably another motor protein down here that wants to walk toward this plus end. And if you have enough overlapping microtubules with enough of these motor proteins inside them, this is just creating a lot of force pushing these spindles apart. And of course, once it gets to the end, there's no more force, there's nothing more for it to do. But luckily, these microtubules are always growing. So as the spindle poles come apart, the microtubules grow more and they provide more length of microtubules for these motor proteins to walk on. And plus end, the most common plus end motor is called kinesin. And this is just a word for force because it's a protein that can generate force. Interestingly, this Pac-Man mechanism also goes through some motor protein mechanism. And there's other proteins that want to go this way to the minus end, including the motor protein called dynein. So, right, there's different motor proteins, and this is, so dynein is a minus end protein, whereas kinesin is a plus end protein. So these different motor proteins go to different places and do different tasks in order to do the job of moving these chromosomes from the middle of the cell, from this metaphase plate of the middle of the cell, all the way to the extreme ends of where the daughter cells are. And here, I hope this movie works out pretty well. No? All right. Okay. The first one worked fine though. I'll try to get that. I'll try to include that for next time. Okay, so we've reached the end. So chromosomes have moved, and now we're at the final phase, which is telophase. If you remember telomere, the end of the chromosome, telo always means end. So here, telophase is the end of mitosis, the final part of mitosis. And I always felt, okay, telophase is kind of just added on to the end. It's not really exciting because the chromosomes are done moving. But actually some pretty interesting things are happening. So chromosomes have to decondense, and more interestingly, this nuclear envelope has to reform. So telophase is here, and you can kind of tell the difference between telophase and anaphase. Can anybody see what the difference is? So this one would be anaphase, and then telophase. And I believe that this one's also pretty telophasey. The difference is that the chromosomes have started to decondense. So these condensin proteins that have been on the chromosomes and making them really rigid and separate from each other. You can see they're really nicely separated rods. Here in telophase, they're starting to get very fuzzy. Okay, so the loops that condensin has brought chromosomes into have started to come apart, and the chromosomes are fuzzing out again and no longer very well separated. So, okay, that's not really too interesting, but the one kind of mystery that's left, though, there's a mysterious part of telophase which is reforming the nuclear envelope. So in organisms like us, where nuclear envelope breaks down, all the little pieces of the nuclear envelope, which when the nuclear envelope breaks down, it actually completely comes apart into these little membrane vesicles, like little tiny soap bubbles. Still, they have some of the nuclear core proteins are still left inside them, and other nuclear proteins, but they just kind of dissociate into these tiny vesicles. and then at some point they all have to come together to reform one single nuclear envelope. And so they have to all sort of nucleate on the surface of these chromosomes. So whatever bit of chromosome ends up on the surface of this blob of telophase chromosomes has to do the job of recruiting these soap bubble vesicles of nuclear membrane. And sometimes, even though I'm showing you here that, it's just one single blob here, but sometimes one chromosome may be late. One chromosome can be slow in getting to the pole, or things start to decondense before the chromosome is already there, and you often end up with a chromosome that's out of the picture, and the nuclear envelope may reform around all the chromosomes except for one, and that can actually be a problem because now that chromosome is cut off from everything else, and its activity, it's almost like you've lost that chromosome, and that can be a serious problem. And in fact, a lot of cancer cells, these are cancer cells here, have this problem with micronuclei. But most of the time, cells don't. They can completely reform the nuclear envelope, and we don't really know how or whether the cell checks to see to make sure that all the chromosomes have gotten in there or not. So. Even though telophase is kind of just the end of mitosis where everything is going back to interphase, there's still some pretty important checks that have to happen in order to make sure that the next interphase is working. And just as a kind of aside, as a consequence of what's happened in mitosis, so you can see that so in anaphase, chromosomes are brought to the poles, and they have this kind of, they're pulled by their centromeres, and the rest of the chromosome is kind of lagging behind. And that's pretty visible in this guy, right? So all the centromeres are here, and the rest of the chromosome is kind of lagging behind. And then in telophase, it's forming the same, it has the same sort of structure. So that leads to this pretty interesting phenomenon called the Ravel orientation for this scientist named Ravel who discovered it or who predicted it. And it means that in interphase, even after the chromosomes decondense, they keep this kind of orientation where all the telomeres, all the ends of chromosomes are on one half of the nucleus, and all the centromeres are at the other end. So especially in cells that are dividing quickly, they don't have a lot of time to re-randomize this their orientation. The cell still has this large-scale organization where chromosome ends are all in one half of the nucleus, and the rest of the chromosome centromeres are in the other half. And that has some pretty interesting consequences that we'll talk about in the next class. Okay, so now I just have a little quiz, and I just want anybody to guess what stage, which of the mitotic stages you're looking at here. Anybody tell what this one might be?
Chromocytes?
Yes, you got it. Yeah. And yeah, I threw in. It's kind of tricky because you might think this is telophase, right? You've got nuclear envelopes reforming here. So blue here is nuclear envelope, green is microtubules, and red is the DNA, of course. You may think, when I first saw this, I thought it was telophase until somebody reminded me that if it were telophase, you would see the chromosomes would all be pointing toward the poles. But it just happens that this cell has two nuclei in it. The cell did not, in the last cell division, it didn't pinch off the two daughter nuclei. And that happens sometimes also, especially in cells that are growing in a dish or cancer cells. So a lot of biology is done with people who grow human cells in a flat petri dish, right? So human cells are supposed to be in tissues in a three-dimensional organization. But we've learned, a lot of what we learned has been growing cells where they're really, really flat. And a lot of what we learned is probably wrong because cells behave very differently when they're flattened out. It's like they have a constant force pushing down on them. And things that are sensitive to forces like the spindle or like mitosis can often misbehave. Okay, so yeah, that's prophase, and that cell just has two nuclei in it. Okay, so there's another mystery here, which is so if those movies had worked out, and I have another couple of movies that I hope are better than the ones I've shown so far. But there's a big problem here. So I've kind of showed the stages as if they're just proceeding from one stage to the next. You've got prophase, metaphase, anaphase, telophase. But the change between metaphase and anaphase is really a single instantaneous change, right? Metaphase, all the chromosomes are at the middle, and anaphase, suddenly all the chromosomes come apart at the same time. And the question is, how do they know when it's the right time? Right? You've got, each chromosome is a unit unto itself. So one chromosome is here at the middle, and at some point it divides, but it is coordinated with every other chromosome, so they're all dividing at the same time. And you can think, if they didn't do that, or if the cell division starts while one chromosome hasn't yet made it to the middle or isn't connected to both sides of the spindle, then again you can get a chromosome loss. You can get something like this where one chromosome is late and the rest of the chromosomes are ready to divide. So the reason why cells are able to deal with this problem and they're able to wait until all chromosomes have made it is that there are several different redundant mechanisms called checkpoints to ensure that the cell doesn't divide until all the chromosomes are ready. And so the checkpoint that we're talking about is called the spindle assembly checkpoint. Okay. So the reason people first were aware of this checkpoint, I mean, just theoretical considerations first made people realize, of course, there has to be such a checkpoint because all the chromosomes could potentially divide on their own, but they actually wait. They wait until they're all ready. Some scientists took some recording of mitosis in live cells. So they showed that even though there's a lot of variability in how long it takes for all the chromosomes to come to the middle. So if you're looking at cells when metaphase has started, because it's a random process, this capture of chromosomes by the spindle is random. Some chromosomes may be ready to divide in an hour. Some cells may take 2 hours. Some cells may take 3 hours for all the chromosomes to get to the center. But as soon as all the chromosomes are in the center, well, not as soon as, but about 20 minutes after, then the cell divided. Okay. So in other words, this is time here. So let's say this is divided, and this is still metaphase. So here's anaphase. So one cell may take a long time to reach metaphase. I don't know if this would make sense. But then it takes 20 minutes to get to anaphase. Right? So let's say this distance is 20 minutes. And it doesn't matter how long it takes the cell to reach this point, this time to go to anaphase is always 20 minutes. Okay. Right. So somehow the cell is able to know, okay, all the chromosomes are at the middle, and now it's time to divide. And the final point that made people realize that actually chromosomes are communicating when it's time to start cell division is if they found a cell where all but one chromosome is at the middle, okay, so let's take a simple cell with only 3 pairs of chromosomes. So let's say 2 chromosomes are at the middle, ready to go. We have a spindle. So these guys are ready, but unfortunately there's a lagging chromosome that's not caught by the spindle yet, right? So from previous videos, we know that once this does come to the middle, then 20 minutes later the cell is going to divide. But if instead of waiting for it to get to the middle, if you destroy it with a laser, or in fact, if you only destroy this, if you just destroy the kinetochore, the center of this chromosome, you destroy that with a laser, then 20 minutes after you destroy this, then the cell divides. Okay. So what that led people to believe is that centromeres, or the unattached centromeres of a chromosome, are sending this signal to the cell that says wait. Okay. If the chromosome is not attached, it's actually sending this unattached kinetochore is sending a signal to the entire cell that says, don't divide yet, I'm not on the spindle. Okay? Because once this gets attached, then the cell divides. Or if you destroy this, then you destroy the signal and the cell divides. So even a single chromosome without an attached kinetochore can make the entire cell wait. Okay? And here, okay, this movie is kind of working. It's going to loop, but this is, I mean, you can't really tell that it's 20 minutes after, but once all the chromosomes are together, they all come apart at the same time. Okay, that's kind of all that movie is showing you. All right. So how could one kinetochore be doing this? So I told you earlier in the class, and maybe last time too, that in addition to just being a handle that the cell is pulling the chromosome on, the kinetochore also contains just a huge number of proteins that are involved in signaling and communication. So yes, there's a surface for microtubules to come in and bind and pull on the chromosome. But those proteins are kind of a small part of the story, and over the past decades, people have discovered a huge network of other proteins sitting here at the kinetochore doing various tasks. Each little, so in the kind of iconography of scientific papers, a protein is usually ellipse or a square of a certain color. So with a label in it. So every one of these is a protein. Every one of these could be a complex of many proteins, and these are familiar. I hope these are the histones. So there's special histone proteins at the centromere, and then there's a huge array of other proteins doing various things. So yeah, I don't even see the kinesin, the motor proteins here. They've left those out, I guess. I guess this is a motor protein, but it's not for going on the spindle. Anyway, there's a huge number of proteins at the kinetochore, so there's definitely some ability for them to signal to the entire cell. Okay, but still, the question is, how does this kinetochore know if it's attached to the spindle or not? And I've given some hints here because we know what happens to the kinetochore once it does get attached, right? So, can anybody come up with a hypothesis of anything different that may be happening to a kinetochore when it's attached versus unattached? Well, let me try to draw a little bit more realistic what this is going to look like. And that might give you a better idea. Here. Here's one that is attached. this picture also shows an illustration of what's happening when kinetochores are attached, right? Remember I said each little kinetochore is being pulled apart from its sister kinetochore. And yeah, so that's the main difference between an attached and an unattached kinetochore. If a chromosome is attached to the spindle, then these kinetochores are pulled apart from each other. So somehow this being pulled apart can be sensed. Because if you can imagine some proteins here, let's say this is in the state of being pulled apart. If it's not pulled apart, then maybe these proteins can interact with these proteins down here, and they're kind of close together. But once it's pulled, once this is under tension, then you're separating groups of proteins that no longer can interact with each other. That's kind of a very simplified way of saying what's going on. The actual mechanisms are a lot more complicated, but it's actually this pulling of the kinetochores apart from each other that's changing the location of some proteins relative to some other proteins, and that's changing the whole biochemical signaling network that's happening at the kinetochore. So just this geometric constraint, whether you're pulling on it or not pulling on it, you're changing the geometry and changing the position of proteins at the kinetochore that will change what kind of signal is being sent. And the way this was shown, the way this was actually proved, is actually pretty cool, and it kind of is in keeping with my theme of trying to find unusual chromosomes and how people found the right kind of chromosomes to use to answer the right question. And it turns out that this question could be directly addressed by looking at chromosomes of praying mantis. We're getting into a little bit of interesting facts about sex chromosomes here. Sex chromosomes are really, really variable. Humans have pretty boring sex chromosomes, right? There's either X or Y, and XX females and XY males. But there's a lot of variations, and praying mantis is kind of the next level up. So praying mantis has three different sex chromosomes. A Y chromosome, just like ours, and if you have a Y chromosome, you're a male. But there are two X chromosomes that are different. They're X1 and X2. They both behave as sex chromosomes. So males have a single one of each of these sex chromosomes. So they have an X1, an X2, and a Y. Whereas females lack a Y, but they have two of each X chromosome. So in meiosis, when a male is making sperm, it either makes a sperm with a single Y chromosome, or it makes a sperm with an X1 and an X2. Right? So if this sperm with X1 and X2 unites with a female egg, when this sperm unites with an egg, it makes a female, and when the Y chromosome unites with an egg, it makes a male. But how do these guys divide from each other? So the X1, the X2, and the Y chromosome have to pair up and divide and go into opposite cells. I'll talk a little bit more about that in the meiosis section. But right now, what we have to think about is that there's a situation where you have three chromosomes. There's an X1, an X2, and a Y chromosome. And these have to be all stuck together and divided into into opposite cells. And this is really tricky, right? For three chromosomes to come together and go to opposite directions. And so, because it's a weird system, sometimes in males, one of these chromosomes takes a really long time to attach. So this X1 or X2 may come off, and there you have a situation where you have a single unattached chromosome, and that will delay the entire cell cycle. Okay. And this is what this looks like. It's really hard to see, But if you were actually looking at it through the microscope, you would be able to tell because you can see them, you would be able to see them moving. But what this is here's the X2 chromosome and the Y chromosome, and the X1 chromosome is down here. And this is where they started recording this movie at time 0. And you can see all the other chromosomes are lined up in the middle of this cell, and they're waiting. They're waiting for the X1, X2Y, and the X1 to line up. Okay? And so here at 23 minutes, they've moved a little bit, so they've been captured maybe by the spindle, and it's coming down. But it's taking a long time for them all to come together. Finally, at 143 minutes, it looks like the Y, X2, and X1 are all together in this kind of arrangement. And then finally, an hour later, so in this case, it takes an hour after the attachment, the cell can divide. So they've waited 3 hours for the sex chromosomes to line up. But since these cells are so big, it's easy to, what people realized is that you could use a very tiny glass needle and actually reach into the cell and pull on the chromosome. So after the X1 and Y got attached, but the X2 is still, this is in a different focal plane. The X2 is a bit lower down, so it's in a different focal plane. They reached in with the glass needle. It's a little difficult to show. I think this is the glass needle right here. You can kind of see the shadow of this needle coming in like that. They pulled it, and as soon as they pulled on it, maybe about an hour later, then all the cells divided again. And the control cell where they didn't pull on anything still took about the same amount of time as last, well, even longer this time. That looked like it took 6 hours. This time it was only 3.5 hours. It can take a different amount of time depending on how long it takes for that final chromosome finally to get to the end. But if they pulled on the chromosome, they could stop this signal from being sent. So it's just like, just like this case of pulling these kinetochores apart. Just by pulling on one chromosome, you could distort the geometry of that chromosome's kinetochore in a way that shuts off that signal. And so this pulling experiment, of course they did it many, many times and showed that the time was drastically reduced. Showed that it actually was this tension. The chromosomes had to experience tension to turn off this weight signal. And then the molecular basis, what proteins are actually involved in sending this weight signal, is still a pretty active area of research. So this tension business is actually one more kind of wrinkle in it, and that is I've shown you different ways that the spindle can pull on the microtubule, but there's actually a few more, right? So there's a little table that tells you whether or not tension is occurring. Of course, if you're pulling on both sides of the kinetochore, then yes, you're getting tension because you're pulling them apart. If you're only pulling on one kinetochore, then obviously not, because if you pull on the kinetochore, the other one is going to come along with it, so there's not enough tension. You can also have what's known as syn-telic. These words don't really, I never remember these terms anyway. If you're pulling on both kinetochores from the same side, that also does not create tension. So even though these kinetochores are usually facing in opposite directions, if there's any flexibility, you might have both kinetochores attached to the same spindle pull. And that could be a disaster because you're pulling it all the way over here to a place where it's never going to be encountered by the other spindle. But the cell will wait until finally that chromosome comes back to the middle. But the last kind of tension is actually the most dangerous kind. And that's when one single kinetochore is being pulled by microtubules from opposite spindle poles. And you might think that, well, there's really not enough room. How could this one kinetochore be pulled by microtubules from both sides? But this is only showing you a single section through this chromosome, and the kinetochore is actually it can be bigger than this, and if you think of this kinetochore as a three-dimensional plate like this, you can have microtubules coming from one spindle and also microtubules coming from the other spindle pole. It doesn't happen that often, but when it does happen, it can be a disaster because as long as the other kinetochore is being pulled, then this can generate tension. So this kinetochore is held in place, and the other kinetochore is being pulled. So this can also send the signal that yes, we're ready to divide. Unfortunately, it's not ready to divide because since this chromosome, this sister chromatid, is being pulled by both sides equally, it's not going to go anywhere. So the cell will divide. This chromosome will move toward this pole, but this chromosome will just sit there in the middle and not divide. So it's still kind of unclear how the cell is able to deal with this, but so I'm kind of going to leave you with some mystery here. But this merotelic attachment, it's called, when one kinetochore is attached to both sides, must be sending some other signal, some other kind of signal that makes the cell wait. So even though it is thought to be under tension, there's something about being attached to two sides. Maybe one kinetochore is getting distorted or something by being bent in two directions. It's not really clear. But somehow the cell knows how to wait for this merotelic attachment. So this tension is not the whole part of the story. It's not tension across the kinetochores is not sufficient to turn off this signal. But as you can see in this diagram, the signal is probably a little less than the signal from completely unattached or from attached to both poles or attached to a single pole, because there's a little bit of tension happening in these cases. There's no tension happening in this case, and there's a lot of tension happening in that case. But anyway, in most cases, the cell is smart enough to wait for all of these possible failures of chromosome attachment to the spindle until everything is attached like this, and there's a lot of tension across the kinetochores, and then the signal is turned off. Okay, so that's that was kind of a lot for today. But if there's any questions about this process, you can ask now or as we're leaving. I think we're going to wrap up there.